biologists, Mr. Fott here. Today we're going to be talking about membrane structure and function just a little bit more. First up are the must-knows. We're going to explain how the structure of these biological membranes have a potential to influence the selective permeability of said membranes. Also, explaining how the structure of these molecules can also affect the ability to then pass through this plasma membrane, and describing the process that is going to allow ions as well as other molecules to move again across the membrane. So first things first, the semi-permeability refers to this membrane that, if you break apart the word, it is selective, or semi sometimes it's called. It means that it's choosy. So it's not everything is allowed through, but only certain things are going to be allowed through. To permeate means to move through. So the literal translation is it is selective or choosy about which things are allowed to permeate. Now, there are two different major groupings here. So we have these small molecules. These could be polar or nonpolar here. They are much more easily able to move across. A few examples might be things like hydrocarbons, hydrophobic, our carbon dioxide, and O2 molecules. Those last two in particular are things that we're for sure going to be focusing on, especially when we come back to talk about cell respiration and photosynthesis. Now, in the membrane, it has a hydrophobic core. So at the center, where you have all those squiggly tails, which are hydrophobic in nature, this is going to create a barrier, or it's going to prevent the passage of things that are charged. So things like ions, as well as large polar molecules. So this whole transport of material, there are two main categories. We can either be represented as passive or active. For a moment, we're just going to talk about our passive transport. By definition, this means that things do not require any energy. In the biological world, energy equates to ATP, so there's none of that needed. So for things to move across, you might be asking, well, if nothing is giving it energy to push it across, how is it able to move across then? It uses the concentration gradient. And oftentimes the AP world likes to say it moves or diffuses down the concentration gradient, which means it's going to move from a high concentration, lots of stuff, to a low concentration, much lower stuffs. So a few examples, again, our hydrocarbons, carbon dioxide, oxygen, and water is another one of those examples that at times can kind of be included within this list. CO2 and O2, respectively, are going to be some of the more important ones that we'll focus on. So down below, we can see the diagram that we can, over time, see that the net fusion or diffusion of these molecules, those little yellow spheres, are going to want to move across from the higher concentration, the left side, to the lower concentration, the right side. Now, when things are diffusing across, every individual thing is treated separately. So we are not going to collectively say, well, there's 7 things on one side and 7 things on the other side. They're equal, right? But what are those things? So it's not just that something's there, it's what is the something that's there. So that's what this slide is really about, is that the yellow particles want to be equal on both sides, much like the purple particles, they want to be equal also on both sides. So we can have different diffusion processes that are going on at the same time. Now, in our examples, we're going to keep it relatively simple. We'll talk about 1, maybe 2 things at a time. But in actuality, if you were to look at a real cell, and this is more so into college, and definitely if you're going to pursue the medical career once you get to your doctorate level stuff, then obviously we don't have just 1 or 2 things dissolved in our cells. There are a slew of a whole bunch of different things. But for simplicity's sake, we're just going to keep it more limited to 1, maybe 2 things. We're going to keep it a little more simple. Now, another way that we can have passive transport is that we could use a protein to assist us. So this is referred to as facilitated diffusion. To facilitate means to aid, to assist, to give some sort of some help to this diffusion process. So it's still moving down the concentration gradient from high concentration to low concentration, as we can see by the diagram over here to the right, where we have our triangles and our spheres, but they're going to use those giant purple blobs, aka our proteins, are going to start to factor in. Now, there are potentially going to be two different versions of this. So we can have things like channels, which are basically like open doors. So it is going to allow things, the passage of these materials, to move across in both directions, in and out of the cell. Or it could be a carrier protein in which it is going to physically pick it up and carry it across the membrane. A few things that are some good examples would be things like ions, and other large polar molecules, so such as glucose, like the one on the right-hand side. We can see those blue spheres. That would be a good representation of a carrier protein for glucose molecules. The actual one that kind of gets thrown in here as well are these aquaporins. So this is again a more recent discovery. So in like the last 5 years or so, that these aquaporins were truly discovered. And so these are extra proteins to allow the passage of our water molecules. A specific example where maybe researchers were finding these things and discovering them is something like the kidney, so the renal system and how the kidney functions. The actual function of the kidney is to filter out toxins and materials and kind of concentrate them into your urine. The kidneys lead into the bladder. And though we can use water to then either dilute or concentrate, which gives us different concentrations of urine, either much more diluted, so a clearer, or a more concentrated, the deeper yellow or other tinge of colors and smells. Sorry if that's a little gross. But that is a good example of where we might find aquaporins. Here, as I mentioned, one of our carrier proteins is like the glucose, and at times that these carrier proteins they don't always have to just be a one-to-one where we just let one thing at a time. They can actually work in conjunction sometimes, not all the time, but in this example, we're actually letting two glucose molecules in for the price of one movement. So this protein here, represented by the kind of bright fuchsia or kind of a purpley color, and the glucose molecules, the green spheres. So those are the ones that are going to fit nicely. And notice how it almost has like a little seat for it. And that's oddly specific to that molecule. So these carrier proteins and these channel proteins, whichever proteins we're talking about, it has a very specific, highly specified shape that it is going to allow whatever molecule it is to help with the facilitation across the membrane. It's highly specific. So for instance, if I had a fructose molecule, which is another type of monosaccharide, if that were to try to get in there, it could not because it does not have the same chemical makeup as glucose. So it is highly specific to whichever molecule it is that needs to move across the membrane. The other type of transport, which is going to require energy, is going to be our active transport. So we need our ATP. This is the big reason why our cells are going to have to do our cell respiration and photosynthesis to generate those ATP molecules so that we can maintain our high concentration of vitamins, minerals, nutrients, ions, and so on and so forth inside of our cells. And this, what's special about it, is going to go against the concentration gradient. So it's actually going to move from a low concentration to a high concentration. Again, we want to maintain that high concentration of materials internally inside our cells. There's a few good examples. Sodium-potassium pump is a good example, as illustrated to the right, and we'll look at that just a little second, as well as protein pumps that are proton pumps that we'll talk about when we get, again, to cell respiration and to photosynthesis. So what this can do and why we might need to do this is if we need to highly concentrate specific ions or certain materials, either internal or external here, we can use the energy to push it against the concentration gradient. Because again, natural diffusion wants to go from high to low. We are going against this. This is almost like if you've ever been on like a kayak or like a canoe or something, and you either flow with the river or you flow against the river. And obviously, if you're going to go with the river, it's much easier. You can kind of kick back, relax, maybe paddle every now and then, stir yourself up, whatever to turn. However, if you're going to go against the current, you're going against the flow about what nature's trying to do, then it's going to require so much more energy. You're going to be exhausted after that from all that energy you just spent trying to go against that gradient. And so that is why we need this energy so that we can highly concentrate our sodium, potassium, protons, what have you, to move them across the membrane. One of the things that our body is trying to do is to generate a voltage or electrical current to create certain sides could be a little bit more positive and a little bit more negative on the other side, kind of like a battery, so that it has that electrical current. One side's going to be the positive terminal, one side's going to be the negative terminal. And fun fact, we do actually have electrical voltage that's running through our body. That's a little bit actually how our nerve cell and how they say that you, if you were to get shocked and you got zapped and you're like, oh my gosh, I got this volt got sent through me. Well, that's true, that it's actually messing with the voltage of your nerve cells. That's what you would feel if you did get shocked. And so that's, again, tied in with our nervous system. And so that is one reason why we might need to have certain gradients of sodiums or potassiums to be pushed in one direction or the other. For our proton pump, this is, again, we'll talk about it more in depth when we get to our cell respiration and photosynthesis, but it's going to aid us in the ATP production process, and it could create, notice how we have a lot of those hydrogens on one side and a little amount on the other side. Maybe potentially you can see where we're going with this, pH. If we have a lot of hydrogens, we're going to drive that pH low, versus if we have a little amount of hydrogens, we're going to drive that pH up. So pH balance, this can also play into that. And if you recall that our conditions, our internal conditions, not just temperature, but also pH, play a huge factor into how certain proteins and enzymes are going to function. Again, we need energy to do all of this. Another function we can see as to how these work is that proteins, they don't just work individually. A lot of them, in fact, are going to work with kind of in conjunction with another one. So it's not just one proton or protein, it's another protein, and then a third, and then a fourth. So they can work together like this. They call this mechanism cotransport. And again, if you break this apart, co means together. Like co-workers, you're working together, or cooperating, you're operating together is what you're doing. So these proteins are co-transporting. They're going to transport, for instance, in this example, they want to get sucrose across. However, you can't do it alone. You need something else. So you need those hydrogen ions. This is where you could have active transport, can then push and drive those hydrogen ions across using the proton pump. And then now you have a high concentration of protons on one side, that's the H plus, and then the low concentration on the other side, it's going to want to come back. And then they can bind with the sucrose-H plus cotransporter. So now you have 1 part H plus, 1 part sucrose, and that allows both of those to move across together. Again, that prefix co. Some differences again to kind of summarize between active and passive transport. Passive is going to require little to basically zero energy. It goes down the concentration gradient, which means from high to low. Sorry about the bell. These are things like diffusion and osmosis, as well as facilitated diffusion, and could potentially be with transport proteins. Oppositely, if we have active transport, this is going to, in fact, require energy. It moves against the concentration gradient, which means from a low concentration to a high concentration. These are things like pumps, for instance, ion, like sodium, potassium, or hydrogen pumps, and are involved in the exo and endocytosis processes, which we are going to end on in just a second. So here's a quick summarization in visuals for active and passive transports. These are excellent things to draw these in your notebook. Again, visualizations are huge in biology. So to move things in bulk, we've kind of already talked about this. This is the endomembrane system in action. If we need to bulk, meaning lots of things, transport across the membrane, or if we happen to need things to fuse with the membrane itself, we can then use this endo or exocytosis. Those prefix, endo means enter, exo means exit. So those 2-letter beginnings, en, endo means enter, ex, like exiting, is going to leave the cell. Thank you all for watching. Please let me know if you have any additional questions. Have a fantastic day.